Today we will learn the new topic. We have finished the mitosis division, and today we will do meiosis. Okay, we will do meiosis. Meiosis division. All right. So this division, this is a division which take place in some special type of cell, that is the what we call the germ cell. All right, germ cell, and this germ cell they produce a reproductive cell. Reproductive cells, that is, those are the gametes, a splice cell. No, these gametes are produced by this meiosis division. This division takes place in sexually reproducing organism. Reproducing organism, and in this sexually reproducing organism, the special cell that they take part, and there is a very special phenomenon which take place in sexual reproduction, that is known as fertilization. Okay, fertilization is the that special phenomenon by which the sexual reproduction takes place. In fertilization is, that is, fusion of gamete, fusion of gametes. And gametes are what? Gametes are a special cell which takes place, means two cell combine with their everything, means their cytoplasm mix together, their whatever the chromosomes are there, these chromosomes also unite. Okay, so this chromosome, whatever is the chromosome here in the gamete cell, that chromosome as a chromosome coming from two different cells. So here the total number of chromosome will become double, is it? Whatever the chromosome number here, say here if it is 7, this one also 7, and then in the zygote it will become 7 plus 7 is equal to 14. So by the process of fertilization, the number of chromosome become double, and fertilization is essential in the sexual reproduction. But what we know, that the number of chromosome in an organism of a species that remain constant. Is it? Number of chromosome must remain constant. The chromosome number should not increase or it should not change. If the chromosome number will change, then the character of the organism will change. Right. So as fertilization is essential and this make the number of chromosome double, so there must be some phenomenon which will be reverse of the fertilization, that this there should be one process by which this increased number chromosome should come down to half. By fertilization, it become double, so there should be one phenomenon which will bring down the chromosome number by half. And that phenomenon is the meiosis. So this reducing the chromosome number takes place by a cell division, that is the meiosis. So meiosis is a type of cell division in which chromosome number is reduced to half in compared to their parent cells. Okay, in compared to parent cell, here the chromosome number become half. Here see in the definition, like that meiosis is a special type of cell division that reduces the chromosome number by half, result in the production of haploid daughter cell. So the daughter cell will be haploid in nature. So here what happened, this meiosis always take place, meiosis occurs in diploid cell. Diploid cells where the chromosome number is 2n, two set of chromosome, and they produce meiosis result. The daughter cell cells, that daughter cells are haploid. Okay, daughter cells are haploid. They will have one set of chromosome. And here another thing is, to complete this condition, okay, to reduce the number of chromosome and also to reduce the content of the DNA. DNA content also is very important. Here in the diploid cell, the characteristic DNA content is C. So in the haploid cell that meiosis will produce, they will have the characteristic DNA content 1C. Both the condition must be fulfilled by the end of meiosis. And to ensure that the meiosis is completed in two successive division. Successive division. As there are two successive division, so naturally the number of daughter cells are four daughter cell, because by one division two cells are produced. Again those two cells will divide again. So by the end of this meiosis, we will get four daughter cells. Okay. Another thing, another very special thing is that we know that before a cell divide, Say like what we have learned in the mitosis. Before the cell divide, there is a replication of DNA, that the DNA molecule is synthesized, and so the DNA content become double. So there is a DNA synthesis, is it? DNA synthesis. So what happened? This diploid cells which is going to divide by meiosis, they will have initially, before the division start, initially they have 2cDNA. As it is preparing for the division, so this DNA content will, by the synthesis of DNA content will increase to 4C, double. Okay? And then this, after the first division, what will happen? The DNA content will come down to 2C, and again next division will be there, the DNA content will further decrease because the chromosome number, chromatids will divide, and so it will come to 1C. So here this is the, this is synthesis of DNA. Here is the first division. It is producing the cells with 2C amount of DNA. In the second division, before the second division, if again DNA will synthesize, then again this DNA content will increase to 4C. So before the second division, there is no DNA synthesis. This is second division. Okay. But here there is no DNA synthesis. So by the end of the second division, DNA content come to the 1C, which is characteristics of a haploid cell. So characteristics of haploid cell is that one n number of chromosome, n number of chromosome, and C, 1C amount of DNA. This is the characteristics of haploid cell. Of haploid cells, or it is, if it is gamete. One another important thing, when and where the DNA, this meiosis occur. Say this is slightly different in plant and animal cell. What is the difference? In case of animal, in animal, meiosis occur, meiosis takes place during synthesis of sorry, during production of gamete. Okay, when there is gamete production. So this meiosis that takes place, it is known as gametogenic meiosis. And this during the production of gamete means we call gametogenesis. This process is gametogenesis. Okay, and the type of meiosis is called gametogenic meiosis. But in case of plant, in plant it is not during the production of the gamete. This is during the production of spores, that is the microspore and megaspore. So in plants, meiosis takes place during production of spores, and this is we call sporogenesis. And this meiosis is called sporogenic meiosis. Genic meiosis, which takes place during the process of sporogenesis. And what are these spores? This is, that microspore which is in the male, that is known as pollen grains, microspores or pollen grain. Right. And there is female that spores, which will later on produce the female gamete, that is the megaspores. Okay, this is called the embryo sac. It produces megaspore pollen grains. Later on, pollen grains is haploid. So pollen grains later on will produce male gamete, and megaspores later on will produce the female gamete or the egg. Okay, so this is the two main difference that the meiosis occur in plants and animal. As such, you can say that it is during the gamete formation. There is in some of the lower group of plants, where the plants are, say, like in algae. In many of the algae, the meiosis occur in the zygote cell, because those plant like Spirogara, okay, they are haploid plant, and so during the gamete formation they will not produce the meiosis. So they directly, their cell function as a gamete. After fertilization it produces zygote, and meiosis occur in the zygote. So that is called the zygotic meiosis. Zygotic meiosis. Okay. Now let's say, so this meiosis, there is, you can, some important point that this meiosis ensure production of haploid phase in the life cycle of sexually reproducing organism. Fertilization make the diploid phase. So fertilization and meiosis, these are the two very, very important phenomenon takes place in sexually reproducing organism. Animals, meiosis occur during gametogenesis. In plants, it is during the formation of haploid spores, during the formation of pollen grains. That is why for the study of meiosis in plant, we collect the anther, very young anther of flower from the flowers. They say here is some key features of meiosis, that meiosis involves two sequential cycle of nuclear division. There are two nuclear division, meiosis 1 and meiosis 2, but only one cycle of DNA replication. DNA is replicated once, but the nucleus is divided twice. In mitosis, one DNA replication and one nuclear division. Okay, meiosis is initiated after the parental chromosome have replicated to produce identical sister chromatids in a space. Meiosis involves pairing of homologous chromosome. This is a very special thing that during the meiosis occurs. Okay, and at the end of the meiosis, four haploid cells are formed at the end of the meiosis 2. Four haploid cells. Meiosis is that there are two successive division, and we call them meiosis 1 and meiosis 2. And each division has, like in mitosis we have learned, prophase, metaphase, anaphase, telophase. So for meiosis 1 there is prophase 1, prophase 2, meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. This 1, 1, 1 is indicating that is the first meiosis. And like that in the second meiosis, the same phases are there, but it is indicated by 2. So prophase 2, metaphase 2, anaphase 2, and telophase 2. What happened in prophase? Say in the mitosis in prophase, what we have seen, that prophase begins with the appearance of the chromosome. When it first appears, that is very, very thin initially, but as the process, the division progress, gradually the chromosome become very clear because they become short and thick. And this shortening and thickening of the chromosome takes place due to condensation of the DNA molecule, chromatin thread. This is, see here, we have the diagram already drawn. Here is, I am showing one cell with a nucleus. This one is the nucleus. This is cell membrane. This is cell cytoplasm, and in the center is a nucleus. Nucleus is having nuclear membrane. This is outside. If I see this is the nuclear membrane is there, and within the nuclear membrane, the threads, you can see that blue and red thread like tangle mass of thread is there. That is the chromatin network. This is chromatin network. Okay, and this body, this white body ball like another structure, this is the nucleolus, and it is bounded by nuclear membrane, and the site of protoplasm within the nucleus, that is what we call nucleoplasm. So together all these structure together is nucleus. So this is, we are showing a phase cell in interphase. This is interphase. Okay. So in the interphase, cell is there within the nucleus. We cannot see the chromosome. And in the animal cell, it is that a prominent structure, that is centriole or centrosome. Centrosome is clear, very near to the nucleus, but it is in the cytoplasm. And this one we know from the mitosis, that this centrosomes are made of centriole. Okay. And from one centriole, two centrioles are produced. That is, it is replicated during the S phase. So we will see this one now. We will describe from the prophase. This is interphase. Say here all these G1, S and G2 has been completed. So naturally it is going to divide. So it has completed the S phase. During S phase, the DNA has been replicated. After that, G2 phase, some protein, RNA, and other things have been synthesized. So this is interphase. After that, prophase start with the appearance of chromosome in the form of very thin thread. And here initially these threads are very long and somehow it is clear. But actually when we can make out that there is a chromosome, then only we can say that the mitosis has started, and it has started with the prophase. Prophase, first prophase of the meiosis, that is very complicated and important, and it takes more time. So prophase is very complicated, time taking, and having some important phenomenon here. So to describe the prophase first of the meiosis first, this prophase has been divided into several substages, and these substages are leptotin, zygotin, pakitin, diplotin, and dikinesis. Okay. So prophase first has been, this is prophase first. Prophase first has been divided into leptotin, zygotin, pakitin, diplotin, and dikinesis. Now, what is the leptotin? Leptotin is the stage of the prophase when we can first see the chromosome. So, chromosome appear as a thin thread. Chromosomes are visible as thin thread like a structure. By this time, nuclear membrane, nucleolus, they are very clear. See here, one thing I should tell you, that what I have drawn here from the prophase, I have drawn only the nucleus portion. All right. The entire cell I have not drawn, because all these, first we are taking the nuclear division. So this one is the nuclear membrane. Okay, nuclear membrane. So this is the nucleus. Within the nucleus, we can see that this thread. Here in the interphase, we cannot make out chromosome, but here it is coming as a thin thread. and the nucleolus is attached with one of the chromosome. So when it appears, then it is interphase. After that, as the process progress, with the progression of the division, condensation also progress. That is further condensation. Then what will happen? This thread, this chromosome will fold more, and then this will be thicker and naturally shorter. Say if I have a thread long this thread, we cannot see properly because it is thin, but it will be long. But this one, when it will fold, then this will be only this much of length, because due to folding its length will decrease, but thickness has increased, so it will be clearly visible. So this is what we call the condensation. So with the progression of the prophase, the condensation also progress, so the chromosomes are now clear more. And here one important phenomenon takes place, that here you can see, that is the chromosomes, two chromosomes which were seen initially scattered, now these two chromosomes has been paired. They have stayed side by side. This is that is the pairing of the chromosome. Pairing of chromosome. This pairing of chromosome is called synapsis. And those chromosomes which pair together, they are actually homologous chromosome. So the homologous chromosome pair lengthwise. Homologous chromosome means that the chromosome which have their morphology similar, the type of gene they are same, their sequence also same. So they are homologous, means similar with their arrangement, number of gene, type of gene. So this is homologous chromosome, and it pair, this pairing is called synapsis. For the pairing, a special type of protein is involved here, and that is that structure, the protein which help the pairing of the chromosome, they are called synaptonymal complex. Synaptonymal complex. Okay. So synaptonymal complex help in pairing of homologous chromosome. Pairing is very specific. It takes place in various ways. It may start from one end and move to the other end, sometime like the zipper chain. Okay, so this pairing completes. This stage is known as zygotin. Zygous means pair. Okay, here again the chromosome you can see in the drawing, it is thicker, so it is more clear. Next stage is that chromosomes are even shorter. Here, of course, in my drawing, just to show you very clearly, I have drawn the nucleus bigger and chromosomes also bigger, but it is thick. So what happened? Gradual condensation, chromosomes are more thick and clear. Now we can see in each of the chromosome, two chromatids are clear. You can see, no? In each chromosome, two chromatids. Now, so altogether here in this paired chromosome, there are four chromatids. This one, this paired chromosome, this is called the bivalent. Bivalent. Bi means two, paired chromosome, two, valence. And here there are four chromatids in a bivalent. This is also bivalent, but there are four chromatids. So this condition is known as tetrads. Okay. So in a bivalent, there are four chromatids. Now, this stage is called packaging. Here, one thing happen. This is known as crossing over. This say you can see this pink chromosome and you can see blue chromosome. Pink chromosome has two chromatid, blue chromosome has two chromatid. So the chromatids of the same chromosome, they are sister chromatids. But relationship between the blue chromatid and the red pink chromatid, this is they are non-sister chromatids. Okay. So this homologous chromosome now they exchange a part of the chromatids between them. There is a exchange, means some portion of the pink chromatid will go to the blue and blue chromatid will come to pink. This is exchange of chromatid. This one is known as crossing over. This is very, very important phenomenon, okay, in sexually reproducing organism. This crossing over. This crossing over phenomenon is so important that this provide variation in the organism of a spring of sexually reproducing species. So crossing over takes place, and as crossing over to take place, this crossing over, this chromosome chromatids has to come close to each other. That is why this pairing, synapsis. After that, this exchange has taken place. Now, this chromosome means what was the function for that exchange? So they came close to each other. That is, they paired. So exchange has taken place. Now, no need to stay together. So these two paired chromosome from the bivalent, they start moving apart. Okay. When they start moving apart, this stage is called diplotin. When it is moving apart, but we can see that the chromatids, non-sister chromatids, are still attached at certain point, and this point indicate where the exchange of segment, that is crossing over, has taken place. This is this structure, this cross-like structure, is it? It is like a cross. This cross-like structure where the exchange has taken place or crossing over has taken place, that one is known as chiasmata. So, chiasmata are the point X-like, cross-like point which indicate the point of crossing over. And here you can see these now the chromosomes are again short and thicker. Okay, so this is diplotin. Now this nuclear membrane is gradually disintegrating, and this one, this is the nucleolus. Nucleolus also has started this disorganizing. Centrioles have divided and started moving to the opposite pole. Here they have started moving, and we can see around them there are some filaments like a structure. This is astral rays and spindle fiber started. And then one step, it is called the diakinesis. Here one phenomenon which take place, that is known as terminalization. Terminalization means this chiasmata that gradually slip towards the end of the chromosome. In the bivalent, as it is moving apart the bivalent, they they are this uh, the chiasmata, they are moving towards the end, towards the terminal side. So this is known as terminalization. And there is nothing else. So this lead to terminalization, lead to this separation of the homologous chromosome, bivalent. Okay, by the time now, by the time that diakinesis has come, this nucleolus has disappeared almost. Nuclear membrane disintegrated, and spindle fibers have moved to the opposite pole. Of course, in case of plant cell, there is no centriole, so we cannot say that centriole have moved to the opposite pole. But by this time, by the end of the prophase, the spindle fiber has started forming. Chromosomes are further thicker and shorter. And this is the metaphase. Now in the metaphase, what we can see, that the bivalent homologous chromosome, they are at the equator. This is the equatorial plane, and homologous chromosome, this bivalent, we can say bivalents have oriented themselves along the equator. A spindle fiber has completely formed. Okay, and the chromosomes, they are held with the attached to the spindle fiber. In mitosis, one chromosome each, that is along the equator, but here the paired chromosomes are along the equator. The bivalents are oriented along the equator. This is metaphase 1. And then this, after that, the homologous chromosome from the bivalent, they have started moving apart. Clear? But in this case, the chromosome has not divided. Still in each chromosome, two chromatids are there. And these chromatids, they are showing this change color. It is indicating the crossing over, exchange of the segment. The red one, pink one, it has joined to the blue, and blue portion joined to the pink. So there is exchange has taken place. So now this chromosome from the bivalent, they are the homologous chromosome. So from the homologous pair, one goes to the one pole and the another one goes to the other pole. This is anaphase. So in meiotic anaphase, the first anaphase, the homologous chromosome move to the opposite pole. Homologous chromosome, and each chromosome with two chromatids. Now, in the telophase 1, by telophase 1, this homologous chromosome, they have come to the poles, and around them the nuclear membrane has started forming. So this is, and then cytokinesis. So two cells with nucleus, two nucleus has formed. Say, one thing here, I have started with one pair of chromosome in a cell that is undergoing division. Naturally, the cells has many pair of chromosome. Like in case of P, there are 7 pair of chromosome. In case of onion, there is 8 pair of chromosome. Okay, 8 pair of chromosome. So this is the so here for drawing convenience, no, I have taken only the minimum, that is one pair. So initially each cell that was undergoing the division, they had uh, only two chromosome, but after the first meiosis, okay, the nucleus, the cell has formed, they are having only one chromosome, but still each chromosome is having two chromatids. Okay, this is that cytokinesis has taken place. So first division completed. After the first division, there will start meiosis second, meiosis 2. In the meiosis 2 also again, what will have? Prophase 2, then metaphase 2, anaphase 2, sorry, anaphase 2 and telophase 2. 2 and then cytokinesis. Cytokinesis has taken place after the first division also. All right. There is from first meiosis to the second meiosis, there is no DNA synthesis. So here what happened, that the interphase is very short, and the from the telophase you can say that from telophase 1, the cell will directly go to the prophase 2, that is to the second division. Almost no interphase. After cytokinesis, directly it is here. So again in the prophase 2, what will happen? There is chromosome condensation, appearance of the chromosome. Each cell, this one will go to the meiosis 2, in prophase 2. This one also separately will divide into prophase 2. Okay, prophase 2, then metaphase 2, anaphase 2, telophase 2, and then cytokinesis. Okay, what will happen here? What is the difference? Say meiosis 2 is just like mitosis division. There is no difference. Okay, so whatever we have learned, that is true for the meiosis 2. Mitosis. So meiosis 2 is a equational division, or it is simply like mitosis. So here in the prophase, the chromosome will be with 2 chromatids. Then metaphase, the chromosome, they are aligned to the metaphasic plate. Anaphase 2, here now in the anaphase 2, the chromatids, sister chromatids will separate like the mitosis anaphase. Okay, sister chromatid separate. This chromosomes make the metaphasic plate, metaphase plate, that they are spindle with the attached with the spindle fiber. So here the sister chromatid separate, chromatid separate and move to opposite pole. And in the telophase, the sister chromatids, which we can call them as a daughter chromosome, they have reached to the poles, and then cytokinesis has taken place. So what will happen? This cell by the second meiosis divide into 2 cells, and this cell also by second meiosis divide into 2 cells. So ultimately 4 daughter cells will produce. Okay, one thing here in this case, that mitosis, meiosis first and meiosis second. Meiosis second is a short process of the cell division. There is not that very long time. Okay, and here the chromatids, they separate initially in the meiosis. First meiosis, chromatid does not separate, is it? Chromatid does not separate. So 4 cells are produced. Here you can see one thing, that these chromatids with some parts from the other chromosome. So what will happen, that 4 daughter cells they will produce. This 4 daughter cell will be with a different combination due to this crossing over. I will take 5 minutes more, okay, and I will show you one video of the meiosis, so that that will help you to understand the process. Mm. Okay, here, yes, so this is uh, here is that chromosome condensation is going on. Okay, this is the prophase, and each chromosome having two chromatids. Here, this is going on prophase 1. This is pairing of the chromosome, and after that, the crossing over will take place. Say it is going on, the crossing over has taken place, exchange, and that's the color has changed, and now they will go to, after crossing over sister chromatids of each, they will, this is now the packeting stage, that they are separating, and these are the places where the crossing over has taken place, chiasmata. Now this is a metaphase. Metaphase first, the paired bivalent, they are arranged along the equator. Okay, this is the metaphase first. Now this bivalent, the homologous chromosome separating, this is the anaphase first. Anaphase first, so it is the chromosome of homologous chromosome separating, and they are going to the pole. So it is now the telophase, and cytokinesis has taken place. So two cells with reduced chromosome number has taken place. Now each one will divide by meiosis 2. Say here. Now two chromosome started the second prophase. Second prophase, this is the centriole going to the opposite pole, and the chromosomes have oriented themselves along the equator. They are attached with the spindle fiber, and now from each chromosome their sister chromatids will separate. Say separated, so second anaphase. This is the second anaphase going on, and then they have reached to the opposite pole, so it is telophase, and after telophase there is cytokinesis. This is the cell furrowing method. This is one animal cell. Here we can show it. So this is meiosis 1, first two cell divide, then by second meiosis, the at the end of the meiosis we get to, and this process gametes are produced, male and female gamete. This is fertilization, embryo. So this is all right today. So we will stop it here, and in the next class we will solve a question based on this. Okay. Anything you want to ask? Any doubt? Hello? Any question?
No, sir.
Okay, then I will stop the class.
Thank you, sir.
Thank you.
Welcome.